Hello everyone, I am Shirosri Pal and we are discussing this question from GATE examination 2020 from the paper Xl-S, question number 10. And the question states, a 250 microliter of bacteriophage stock containing 8 into 10 to the power 8 phages per ml is added to 500 microliter of E. coli culture containing 4 into 10 to the power 8 cells per ml. The multiplicity of infection is blood. So this is a numerical answer type question. Now let's see what are the parameters that are given in this particular question. So the first thing is the volume of the bacteriophage stock, which is given in the E. coli culture. So this is actually the volume of the inoculum that is added in the E. coli culture. Volume of the inoculum. Next parameter given is this particular volume of the bacteriophage stock contains 8 into 10 to the power 8 phages per ml. So what is this? This is PFU. So PFU is plaque forming unit, or what it means that the number of particles, the number of phage particles that can actually form plaque per unit volume per ml. Next thing that is given is this volume of bacteriophage, the volume of inoculum is given, the PFU of the phage stock is also given, and it is added to 500 microliter of E. coli culture containing 4 into 10 to the power 8 cells per ml. So it actually gives us an idea of the total number of cells. But the total number of cells is actually not directly given. So that is what we have to calculate, because it is stated that 500 microliter of E. coli culture containing 4 into 10 to the power 8 cells per ml. So what is the total number of cells that we have to calculate? And we have to calculate the multiplicity of the infection. So what is multiplicity of the infection? Multiplicity of infection, or which is also called MOI, it is a frequently used term in virology. It refers to the number of virions that are added per cell during an infection. So the number of virus particles that are added per cell during the infection. This number can vary. So it depends on how much we want the cells to be infected, what is the output that we desire, and what is the type of cell. On all of these things, this MOI value will depend. And so it basically means that the number of particles that we add per the number of cells. So basically, if we add 1 million virions to 1 million cells, the MOI will be 1. So if we have 1 million cells and if we add 1 million virus particles, that means the MOI will be 1, because for 1 billion cells, there are 1 million virus particles. Now, for 1 million cells, if we add 10 million virus particles, then that MOI will be 10, because the number of virus particles that we're adding per cell is 10 times. So that is MOI. Now, there is a formula here that you have to remember for solving the sum. That is, volume of inoculum equals to MOI multiplied by total number of cells and whole divided by the PFU. So this is the formula that you need to remember for solving this particular sum and for determining actually how much inoculum to give in a particular culture while you want to do an infection. So volume of inoculum equals to MOI into total number of cells divided by PFU, and PFU stands for the total number of virus particles that are capable of forming plaques per unit volume. So now we will go back to the question and we will find out what are the parameters that are given and we will put them in this particular formula. So first of all, let us write down the formula here again. Volume of inoculum equal to MOI into total number of cells divided by PFU. Right. So first of all, we have to calculate the total number of cells. So it is given 500 microliter of the E. coli culture is given with 408 cells per ml. So 1 ml has 4 into 10 to the power 8 cells. And if we do unitary method, 500 microliters, or which is 0.5 ml. 500 microliter, which is 0.5 ml, will contain 4 into 10 to the power 8 into zero point 0.5 number of cells, which will be 2 into 10 to the power 8 cells. Right? So this is our total number of cells. Right? So volume of inoculum is given 250 microliter. So let us put that here. 250 microliters. Okay. So here the MOI we have to calculate, which is X we are considering. The total number of cells we have just found out, 2 into 10 to the power 8 cells. 2 into 10 to the power 8. And the PFU was given 8 into 10 to the power 8. Now we have to consider the units as well. The PFU was given in 8 into 10 to the power 8 per ml, right? So therefore we have to put this volume of inoculum also in the same unit. So we will change it to ml, right? So we will write 0.25 ml will be equal to x into 2 into 10 to the power 8 divided by 8 into 10 to the power 8 per ml, right? So for the sake of calculation, we are removing the units for now. Now, 10 to the power 8 and 10 to the power 8 will cancel. And this will give us 4. So we're getting 0.25 equals to x by 4, or x will give us 4 into 0.25, which is 1. So we calculate our MOI as 1, right? And that is our answer. The multiplicity of infection is 1. All right, and that's it. Thank you.